Hey, hi everyone. Today we will continue with our discussion of paper 2, and today our sharing will be on MRSM paper, SPM trial paper 2021 paper 2. Okay, let's begin, yeah. Okay, let's look at this question. Number 1. Diagram 1.1 shows sequence of the organization of cell in human. So you have cell P, and then you have tissue Q. Name tissue Q and one system which consists of tissue Q. Okay, let's look at tissue Q. Tissue Q is what kind of tissue is this? This is smooth muscle tissue. Now, why is it smooth? It's smooth because the shape, from the shape, you know that it is a smooth tissue and not a skeletal tissue, yeah? Next, the system that contain tissue Q are, for example, digestive system, blood circulatory system, excretory system, and reproductive system. Okay, next. Diagram shows a cross-section of the arterial wall. Tissue Q form the wall of the artery. Okay, just now we already named tissue Q as the smooth muscle. Explain how tissue Q in arterial wall can lower the blood flow to target organ. So tissue Q is a smooth muscle. So tissue Q can contract to decrease the size of lumen. Hence, the artery become narrower. So it contract to make the lumen to become smaller. So then the artery become narrower. So we say vasoconstriction has occurred. That means the artery has undergone vasoconstriction. So that will reduce blood flow, and therefore blood flow become restricted. Okay, right. Let's see the next question. A blood test showed that the number of cells in individual is below than normal. Explain the possible consequences of this condition on his health. Now, cell P, just now we saw already that cell P is, okay, let's move back. Cell P is red blood cell or erythrocyte. Okay, so what is the condition of the person who has low red blood cell count? Now, first, the person will become very fatigued, very tired, cannot carry out vigorous activity. Now, that's because having less red blood cell would mean having, when the RBC is less, so less hemoglobin in the red blood cell, there will be less hemoglobin to combine with oxygen to form oxyhemoglobin. Okay, so since there are less oxygen, therefore less oxygen is transported to the cell, and the person will have lesser cellular respiration being carried out by the cell. So therefore, he will have less energy. Okay. Okay, let's move on to the next question. Now, question 2. Diagram show a reaction of enzyme X on a substrate. So this enzyme is acting on this substrate. This is the substrate. Okay, now let's look at the product. If this is glycerol, that means the substrate have to be lipid, and the enzyme has to be lipase. And the The other product should be fatty acids. Okay, so Y is a complex form between the enzyme and the substrate. Okay, name X and Y. So X is lipase, Y is the complex of lipase lipid complex, is the enzyme substrate complex. Based on diagram 2, state two characteristics of the enzyme lipase. Okay, now you must look at the diagram and describe the characteristic of the enzyme based on the diagram. Okay, so the diagram shows that the enzyme is highly specific, having specific active site that can combine with the lipid. And the reaction that is shown is reversible. Okay, how do you know that it's reversible from the diagram? Okay, now you can see that the arrow is forward and backward. That means it's reversible. Okay, and the other thing you can see is that the enzyme molecule, the substrate, the structure itself does not get destroyed by the reaction. So it remains the same, unchanged at the end of the reaction. Okay, so how do you know that? Okay, let's move back again. So you can see that the enzyme here, the structure of the enzyme here, okay, it remains the same even after the reaction. It does not change. Okay. Right, next. The medium for enzyme X reaction is changed to pH 3. Explain what will happen to the active site of X. Now, if the pH is changed, enzyme X is lipase, and lipase works best at pH and alkali, slightly alkali condition or medium. So what will happen if you change it to pH 3? Now, the medium that is pH 3 is acidic, and it will change the charges of the active site of the enzyme active site. So the enzyme active site become more positively charged. So the shape of the active site now is no longer complementary, right? Use the word complementary to the substrate. That means it cannot bind to the substrate anymore, or rather the substrate cannot bind to the active site of the enzyme. So the enzyme substrate or lipase lipid complex cannot be formed. Okay. Hence, you will have less product. That means you will have less fatty acid and glycerol produced. Okay, so a medium that is changed to become a different pH, which is not the optimum pH for the enzyme, will make the enzyme substrate less formed, therefore less product produced. And you can also say that the enzyme now has become denatured. Now, enzyme work best at an optimum pH. So for the case here, lipid is slightly acidic around pH 8. Okay, let's look at question 3. Diagram 3.1 and 3.2 show the activities performed by two individuals, S and T. So S is running. So you can see here from the equation itself that you know that there is no oxygen added. So this is lactic acid fermentation, yeah. And individual T, there is oxygen added, so you know that this is aerobic respiration. Okay, so individual S, what is the type of cellular respiration that's being carried out by individual S, and aerobic respiration, or better word will be lactic acid fermentation. Now, individual T, who is resting and reading a book, is undergoing aerobic respiration. Okay, so what is the word equation? We have to complete the word equation for individual T. So we have to fill up the space here. And for individual S. So individual T is carbon dioxide plus water plus energy. So the two missing word is carbon dioxide, water. For individual S, who is undergoing lactic acid fermentation, is glucose, water, and energy. Okay. Okay, please note that for biology, we normally write word equation. See, the question also say you must write in word equation and not chemical formula, yeah? Okay, according to expert running coaches, he said to become a faster and stronger runner, it is important to stay consistent with the training schedule. Based on the statement, explain why an individual S is able to run faster after an intensive training. So we are going to look at what does intensive training does to an individual. Now, first of all, your muscle, if you train often, your muscle is going to synthesize or make more mitochondria. Now, more mitochondria means more cellular respiration. More cellular respiration means you will increase the rate of cellular respiration. Okay, the rate of cellular respiration will increase with more mitochondria. So more oxidation of glucose, therefore more energy is produced. So when you have more energy produced, so you become a stronger runner with more energy to run faster. Okay, next, question 4. Diagram 4 shows a synapse that separates the synaptic knob from the neuron dendrite that receive the impulse. So here is this is the dendrite that is receiving dendrite. Yeah, this is the okay. So you have K there and J there. So what is J and what is K? Now J is synaptic vesicle and K is neurotransmitter. Okay. Parkinson's disease is an example of health issue related to the nervous system. Symptom in Parkinson's disease are due to a loss of neuron that produce a type of K in the brain. Name the type of K involved and state the symptom of Parkinson's disease. Now, a person who has Parkinson's actually has a lower production of dopamine. Dopamine is a neurotransmitter, so that will cause a person to have Parkinson. So the disease, the symptom associated with Parkinson's disease is tremor. So the person will tremor, his hand will tremor all the time, or his limb or his jaw, and he will have difficulty in maintaining his body posture. So he will lose his balance. And he will also walk more slowly, okay? Because he can't maintain his posture. And his speech will change. So he will have slurred kind of speeches, right? and he has rigid muscle here, means he can't have flexibility when he hold something or take something in his hand. His hand become rigid because of a rigid muscle. Okay, explain the effect of painkiller on nerve impulse transmission across the synapse. Now we want to see what painkiller does to the impulse transmission. Yeah. So the first thing is painkiller stop pain in the patient. So we want the transmission to be slower so that the pain signal can be slowed down. So when you take a painkiller, you're actually slowing down the nerve impulse, the transmission of nerve impulse. So the painkiller reduce the release of neurotransmitter. So when you have less neurotransmitter, automatically the transmission of nerve impulse will slow down. Okay, so there's less neurotransmitter being transmitted across the synapse, hence less formation of a new impulse that will generate the pain impulse, right? You will have less pain reaction. Okay, so you can also say that the response becomes slowed down. That means the person respond will be slowed down. Okay, let's move on to question 5. Diagram 5.1 and A and B show two action of antibodies on antigen. Okay, so you have the antibody that's shaped like a Y and the antigen. Okay, this is how it reacts. You can see that the antibody clump together all the antigen. Name the cell in the body that produces antibody. Now, the cell that produces antibody is lymphocyte. So you can also say B lymphocyte. If not, just lymphocyte will do. Name the action of antibody on antigen in diagram 5.1 and A and B. Okay, so let's move backward and see whether you can recognize. Look at the antigen here. It's all broken up. It breaks. That means the process is a lysis, while this one seems to be gathered and clumped together. Okay, so these two processes here, the first one here is lysis, okay, because it breaks up, and the second one is agglutination. Okay, for the A part 1 just now, please remember you cannot just write white blood cell because white blood cell is a general term for all leukocytes. And you learn 5 types of white blood cell. So you have to be specific which is the white blood cell that produces antibody. Okay, next. Diagram 5.2 show an information about Pfizer BioNTech vaccine. Under the national COVID-19 immunization program, 80% of the nation population will be inoculated in the biggest ever vaccination exercise in Malaysia. So I'm sure most of you would have got your vaccination as well. So you have the first dose and the second dose. After the second dose, you receive full immunity. Name the type of immunity obtained by injecting vaccine in the body. So when you inject vaccine, what kind of immunity you get? You get artificially acquired active immunity. Now, artificial because you did not produce the antibody yourself, you are artificially inoculating a vaccine so that it will trigger your body to produce the antibody. So it becomes an active immunity. Explain why an individual is required to take second dose of vaccine in order to complete the immunization. Now, the immunization usually, at least there are 2 injections to complete the whole immunization. That's because the first injection normally is insufficient to stimulate the production of antibody to go beyond the level of immunity. So it just started to produce the lymphocyte, right? So the antibody level is still very low after the first injection. So therefore, you need to go to the second injection in order to increase your antibody production to a higher level so that it goes beyond your immunity level. Okay, normally the graph is like this. So the first injection, if this is the level of immunity, then if you inject the first injection here, it only probably increase a bit and then it come down. So it is insufficient. It is insufficient. The first injection is insufficient to protect your body against COVID-19. So the second injection given here will allow the lymphocyte to be stimulated even more to produce antibody that goes beyond your immunity level. This is the immunity level. So that means your production of antibody is needed to be above the level of immunity. Then only you are said to have gained immunity against the disease, or in this case here, COVID-19 disease. Okay. Okay, let's look at this question now. The community here is show a community how which can form a herd immunity. So you see this close contact, this person is infected with COVID-19, and the person that is close contact to this individual is all vaccinated already. The blue color is vaccinated. The orange color is unvaccinated. So since the blue color are vaccinated, they are protected against COVID-19, although they are very close contact with the person infected. Okay, so here, explain how herd immunity stops the spreading of COVID-19. Okay, so we're going to see now, how does it stop? Now, the vaccinated close contact may not be affected. Even if they get infected, they are not severely infected. So their infection is very mild. So the close contact were less likely to spread the COVID-19 to the unvaccinated people who are close contact to them. So you break the chain of COVID-19. That means this person get infected, he will not spread it to the close contact because the close contact has been vaccinated. So in this way, you are breaking the chain of transmission of COVID-19. So which means that now the whole community become immune to COVID-19. Okay, next, question 6. Diagram 6.1 show a type of response in a plant. So you can see that the plant before and after 1 week, you see the direction of the plant. Okay, in table 1, mark one characteristic of response shown in 6.1. So the answer is the response direction depends on the direction of the stimulus. Now you see here, the direction of the stimulus is coming from a little hole in this box here. So the stimulus is light. So the plant is moving towards the direction of the stimulus. So the plant is responding, the response of the plant is towards the light. So therefore, the answer is the response direction depends on the direction of the stimulus. Based on diagram 6.1, explain the role of auxin in this response. So auxin is a hormone that causes the elongation of stem. In this case here, since the light is coming from one direction. So auxin will move away from the exposed side to the shaded side. So let's say this is the stem. I'm drawing just a shoot. Now the light is coming from this direction. Auxin will always diffuse or move to the shaded side. Okay, this is the shaded side. This is the illuminated side. Okay, so since the light is coming from this direction, therefore the auxin, which is denoted by the dots, move to the left side more, or the shaded side. Since auxin stimulate elongation of shoot, so this section here will elongate faster than this side. Okay. All right. So since the auxin accumulate in greater concentration on the shaded side, so it will elongate. Okay, more on the shaded side, it will elongate more compared to the illuminated side. Hence, the shoot bend towards the light. Okay. So you can say that the should show positive phototropism. Positive phototropism. Okay, let's move on. Plant leaf contain many types of photosynthetic pigment that absorb light and has a color. Diagram 6.2a and 6.2b show two methods of growing flowering plants in greenhouse. So you have one exposed to the LED blue light, and the other one is given fluorescent light. Okay, explain the difference on the rate of photosynthesis between plant in diagram 6.2a and 6.2b. Now, the rate of photosynthesis in 6.2a is faster than 6.2b. Okay, so that means LED blue light provide the optimum light to the plant, so the rate of photosynthesis is faster or higher than 6.2b. So the LED has a strong blue and red wavelength, okay? Now, the blue light is absorbed more by chlorophyll pigment, blue and red light. Usually blue and red light has a higher absorption by the chlorophyll. So therefore, since the chlorophyll absorb a lot of blue and red light, therefore the rate of photosynthesis become faster in under the LED blue light. Okay, next. In March 2011, Japan was struck by an earthquake and tsunami, which caused a nuclear plant to breach and massive nuclear radiation to local area. Okay, so the diagram here, the 6.3, show that sunflower are used to clean radioactive soil in Japan. Based on diagram 6.3, give the definition of photoremediation. Now, photoremediation means the treatment or the method that uses sunflower to degrade, extract, or eliminate radioactive substances from the soil. So the method using the sunflower, the sunflower has the ability to eliminate the radioactive compound in the soil. Explain one example of another plant used in photoremediation. So there's many actually. They want one example. So you can say water hyacinth, which can accumulate heavy metals in water. So since it's 2 marks here, you not only give the example, you need to explain why is the plant that you have suggested, what does it do? Okay, the plant here, you get 1 mark here first for naming the plant, and the second mark is for explaining what it does. So water hyacinth can accumulate heavy metal. So please remember the word explain, you must explain your example that you give. Or you can say water lettuce can treat waste water, or you can say water spinach, which can absorb mercury in soil. Okay, it can also absorb cadmium. So any of these, any pair of answers. Okay, next question 7. Diagram 7 shows 2 plants in 2 different habitats. Okay, so you have plant P that lives in water. You can see the fish there. And plant Q that live above water. You can see that the roots is underneath the water. So state the classification of plant P and Q based on their habitat. So plant P is a hydrophyte. A hydrophyte is a plant that live underwater or live in the water. And plant Q is a halophyte. A halophyte live in a high salinity condition. State one example, one problem faced by plant P in its habitat. Now, what is the problem? Now, living in the water means there's lower dissolved oxygen content, and they also have to put up with water resistance, meaning the water that flows create a resistance to the plant. And the plant that lives underwater, in the water, submerged plant normally receive less light intensity. So any of this problem is accepted. Explain one adaptive feature of plant P to carry out the photosynthesis efficiently. Now, what can he do? Having all these problems that is listed, okay, low oxygen content, water flow resistance, less light intensity. How does he overcome this problem? Okay, one way is to have thin and small leaf. Now, thin and small leaf are less likely to the leaves are less likely to be torn by the movement of water. Okay, if the leaves are very large, then the plant leaves may be easily torn by the moving water. So normally these plants have very, very small and tiny leaves. Okay, the leaves, like for example hydrilla, they are very thin and small. So this will increase the total surface area over volume and increase the diffusion of water as well as carbon dioxide or dissolved gases. So it makes it easier to receive oxygen. All right, remember the one of the problem faced is low oxygen content. So having small leaf actually solve 2 problems here. One is to reduce the likelihood of the plant leaf being torn by the moving wave or moving water, and also increasing the total surface area to allow more diffusion of dissolved gases. Okay, next is having small and hollow stem help the plant to float up, right? This is mainly for floating plant. Floating plant needs to float above water in order to obtain maximum sunlight. And they have hollow stem here, which acts like a float. So they can float upright. Either they can be the one that is found, or even the one under the water too. They have small and hollow stem. Okay, their stem are normally hollow. Okay, so that they can absorb maximum sunlight. Okay, describe the importance of an ecosystem which is dominated by plant Q as a protection zone and fishery resources. Now, plant Q was a halophyte. Okay, so halophyte here are mangrove plants or mangrove trees. So you know mangrove trees grow in mangrove swamp. Mangrove swamp are area that is usually always inundated by seawater, as well as it faces the strong impact of wave and wind. So having mangrove tree that grow along the riverside that faces the sea, okay, so these are all the mangrove trees, okay, that will reduce the impact of the strong wave and wind from the sea. Yeah? So it acts as a protected site for small aquatic organism also. Now in mangrove swamp, there are many types of aquatic animals, right? Aquatic animals that can be protected from their predator by living in this protected site. It also serves as an area for many types of species of migratory birds. So here we are looking at plant Q. Remember how plant Q, what does it do to protect the zone where it is in the mangrove swamp? What does it do? Okay. For fishery resources, mangrove swamp is a very rich habitat for many type of fish, right? So it's a conducive area for fish rearing in floating cages. So this become a source of income for fishermen. So fishermen normally will rear fish in floating cages at the mangrove swamp. Okay, and it's a suitable area for commercial species, various kind of fish breeding. Okay, so that is the importance of the ecosystem mangrove swamp. Okay, we are now to question 8. Diagram 8.1 show the production of insulin by genetic engineering technique. So you can see here, this is a human DNA, and this is the plasmid of the bacteria. You remove the insulin gene as well as the plasmid, and you cut and insert the insulin gene into the plasmid, and then you get this recombinant DNA of the plasmid. Then you insert it into the DNA bacteria. In this way, the clone recombinant DNA, recombinant bacteria will produce a lot of insulin in which you can isolate and purify. Yeah? Okay, so define genetic engineering. So genetic engineering is a technique used to manipulate genes. Okay, you use this technique to manipulate the gene to create new combination of gene in organism. Next, explain how insulin gene is inserted into the plasmid. Okay, so you can see here just now, this is the part here where this is the insulin gene that is inserted into the plasmid here. This plasmid here the gene here, the original gene of the plasmid has been removed and inserted and replaced by the gene that you obtain from the human DNA. So how do you do it? You take the human insulin gene first and you slice it, and you do that by, of course, not with a knife, you use a restriction enzyme. And then you also slice the plasmid, also using restriction enzyme. And then you mix or you combine or place the insulin gene which you have cut into the plasmid. Okay, also by using DNA enzyme. Now, all these enzymes are used for this procedure in genetic engineering. When you slice, you insert, you use enzyme. So the enzyme that you use to cut is called restriction enzyme. Okay, insulin gene from human is inserted into the plasmid. Okay, this bacterium will then be cultured extensively to produce insulin. Based on the above statement, state the disease that can be treated by insulin. The use of bacteria has been suggested in genetic engineering or genetic techniques. Explain about the suggestion. Okay, so the disease that can be treated by insulin is diabetes mellitus. How do you, the use of this bacteria, how do you use this bacteria as suggested in the genetic engineering techniques? Now, the bacteria just now, the recombinant bacteria that we obtained, that has the ability now to produce insulin, it can reproduce asexually very fast. So it can be, that means not only this new bacteria, this new recombinant bacteria are able to produce insulin very fast. So that means it can produce insulin in large quantity, and which we can separate and isolate it from the bacteria, and then we purify it. So this will allow us to obtain large quantity of this insulin at a very low cost and faster. Okay, and the insulin produced by this recombinant bacteria is the same as the insulin that is produced naturally by human. So not only do you able to produce in large quantity, you are able also to produce in at a cheaper cost. Okay, so that means you don't have to obtain insulin from animal anymore, and therefore it is no longer a dispute among people of different religious belief, because this is produced by bacteria, which is acceptable by everyone. Okay, please note that the word diabetes alone is not acceptable. You must say diabetes mellitus, because there's another diabetes called diabetes insipidus, which is totally different from diabetes mellitus. So please do not write diabetes alone. You must say diabetes mellitus. Okay, question 9. Diagram shows a reproduction process in flowering plant, right? So you see this one is a process X, and then there's a bee here pollinating transferring the pollen grain to the stigma of flower Q. So the bee is transferring from flower P, the pollen grain, and the process is of course pollination. And then after pollination, you'll find that the pollen tube will allow the male gamete here, the L, to be transferred to the developing embryo here that has an egg cell and the polar nuclei in the middle here. Okay, so yeah, to explain process X. So process X is cross-pollination because it's from two different flowers, from two different plants. So it's the transferring of the pollen grain from the anther to the stigma of flower Q. So from the anther of flower P to the stigma of flower Q, and the B is the pollinating agent. Okay, compare the reproduction organ between flower P and flower Q. Okay, so let's move back again. So you see that flower P is only have anther and filament, while flower Q has the stigma, style, ovary, and ovule. So one is a male flower, and the other one is a female flower part. So the differences and similarities, because the word is compare. Compare means you have to give similarities. Both produce gamete. Both are located at the flower's organ. Both undergo mitosis and meiosis. Okay, so male flower consists of stamen. Female flower consists of carpel. Male flower has filament and anther. Female flower has stigma, style, and ovary. Male flower produce pollen grain. Female flower produce embryo sac. Male flower project out from the base of the flower, whereas female flower is located in the middle of the flower. Based on diagram 9.1, explain the formation of seed after process X. Okay, let's look at again, back to process X here. So after process X, which is pollination, we want to see what's the event that takes place until the fertilization of the egg cell, until the formation of zygote, yeah? Okay, first you say after process X, which is pollination, the pollen grain will germinate and form a pollen tube. Okay, so then the pollen tube grows down towards the ovule through the skull. Okay, so if you the generative nucleus, okay, so the pollen tube will grow towards the ovule through the star. Okay, so it's like this. Okay, okay, so the pollen grain form a pollen tube. The pollen tube move downwards. Here is the embryo sac. It moves downwards until it reaches the micropyle. Yeah. So inside the pollen tube. Okay. So I'm going to blow this part up here. So the inside there is two nucleus. One is called tube nucleus that lead the way. Tube nucleus is in front, while generative nucleus is at the back, following the tube nucleus. Okay, so then the generative nucleus will move along the pollen tube towards the ovule P4. At the same time, the generative nucleus will divide by mitosis. So you must say that this generative nucleus just now, this generative nucleus will divide by mitosis to form two male gametes. So which means that if eventually this generative nucleus that follow behind the tube nucleus will divide by mitosis to form two male gametes. So you have now three male gametes Okay, you have three nucleus following, moving towards the micropyle. One is the tube nucleus and two male gametes. So you have two male gametes following behind the tube nucleus, because the generative nucleus here divide by mitosis to produce these two male gametes. Okay, the end of the pollen tube will secrete an enzyme then to digest the tissue of the style, and the tips of the pollen tube will burst. So once it reaches the micropyle here, okay, it goes down here and then reaches the micropyle. The end of the, okay, at first it is it's a blunt end here. The enzyme inside the pollen tube will digest, so making it a space channel for the gamete to move into the embryo sac. So the pollen tube, the tip of the pollen tube will burst. Okay, wait now. Okay, the pollen tube then will penetrate the ovule through the micropyle. So it's the pollen tube that will penetrate through the micropyle. The micropyle is the opening at the end of the embryo sac, and then the tube nucleus will degenerate. Having lead the way, now it will degenerate so that it can allow the two male gamete to enter the embryo sac. Now, one of the male gamete will fertilize the egg cell to form a diploid zygote, and the other male gamete will fuse with a two polar nuclei to form a triploid nucleus endosperm. So this whole process is known as double fertilization. Now, double because there's two fertilization that occur. One is P9 here, is where here, this is the first fertilization where the male gamete fertilize the egg cell to form the diploid, and the second fertilization is the other second male gamete will fuse with the two polar nuclei to form a triploid endosperm nucleus. Okay, so this is the whole process of the formation of a seed after pollination. Okay, so after double fertilization, then the ovule will develop into the seed, while the ovary will develop into a fruit. Okay, Mr. John, okay, let's go to question B. Mr. John has planted the watermelon in a cold climate in a greenhouse so that the temperature is maintained at 25 degrees Celsius to ensure that his crops grow well. Unfortunately, the watermelon plants bear very few fruits. According to the agricultural officer who came for a visit, the pollination process by insect rarely occur in a greenhouse. So he suggested another suitable technique to overcome the problem. So diagram 9.2 show watermelon produced by Mr. John greenhouse farm after he applied the technique which was suggested by the agricultural officer. Okay, so you can see here that is the watermelon being produced after he do what the officer suggested. So explain the technique and its advantages. So you look at the watermelon, it does not have seeds. So what kind of technique has been advised by the officer to him? Okay, the officer would have suggested the technique called patinocarpy, because you can see the watermelon does not contain seeds. So patinocarpy method is using phytohormone treatment. Okay, so you spray the flower, okay, the watermelon flower with auxin, and this promote the ovary to undergo repeated mitosis, right? So there is no fertilization, therefore producing seedless fruit. So this watermelon is seedless. So you don't have to wait for pollination, don't have to wait for fertilization in order to produce the fruit, yeah. So the fruit is formed by spraying hormone. Okay, so there is no need to wait for the plant to pollinate and to fertilize. So this is a technique that is faster. It produces fruit without seeds. Okay, so this is a faster technique, shorter time. Okay, next, let's look at this question where the human leg is moved from position X to Y. That means the person is in the leg is in a bend condition to being straightened. So you have S and R muscle shown there. Based on diagram 10.1, explain the function of joint T. Right, joint T there you can see is between the hip and the femur. So it's a ball and socket joint, yeah, which allow rotational movement in all directions. That means between the hip or pelvic girdle with the femur is the ball and socket joint. Okay, so it allows the leg in a to swing in a circular motion. Okay, other example are the shoulder joint, which is between the humerus and the pectoral girdle. So for T here is the hip joint between the femur and the pelvic girdle. Okay, next, describe the action of muscle, bone, and tendon during Y position. Okay, let's see what is Y position. Now, Y position is standing position. That means the leg is from a bending position to a straightening of the leg. So the muscle involved here, because they ask for muscle, bone, and tendon, so you got to mention all three. So muscle always act antagonistically, meaning they act in pairs. So when one muscle contract, the other muscle will relax. So the S is quadriceps femoris. It will contract to produce a pulling force to the tibia. And this pulling force is transmitted by tendon. So remember, muscle always contract to produce a pulling force. And the pulling force, okay, muscle contract. When it contract, it produces force. And the force is transmitted by tendon to the bone, all right, to where it is attached. All right, and at the same time, the other muscle, bicep femoris, will have to relax. Okay, when it relax, because the one muscle will contract, the other muscle will relax. So it will pull the tibia forward and the leg becomes straightened. So you must mention about the antagonistic muscle working in pairs antagonistically. Now, even if you cannot name the muscle, you can just say S or R as shown by the diagram. Okay, and tibialis is just below the bicep femoris. So anyway, this point here, if you don't know the muscle, you don't have to name it. Okay, to bring down the heel. Okay, next, diagram 10.2 show the skeletal system of a pigeon nestling. However, muscle Q in the pigeon nestling is not fully developed in the growth process. So Q is inside, P is outside. So identify muscle P and Q and explain how the abnormal condition affect its movement. So P is pectoralis major and Q is pectoralis minor. Okay. All right. So look at the outside here is major, Q is minor. So when minor is abnormal, his condition is abnormal. All right. If there's an abnormality in the, okay, because the question say here Q is not fully developed. So pectoralis minor is not fully developed, therefore it cannot contract, and because both muscle work antagonistically. So if minor cannot contract, major cannot relax. So which means that both the muscle cannot produce the force to transmit it to the tendon. So the tendon then cannot transmit the force to the humerus, the bone. So therefore the bird cannot lift up his wing. There's no upward lift of the wing, okay? Because the muscle cannot contract and relax to pull the wings up. Okay, so the wing cannot be pulled upwards. No air pressure is there to provide the upthrust, and the bird cannot fly, actually, cannot provide the upward lift for the young bird, and the young pigeon cannot fly. Okay, see, diagram show the movement of a grasshopper and an earthworm. So this is the earthworm movement, this is the grasshopper movement. So you are to give similarity and differences between both. Now, both grasshopper and earthworm have skeletal system, which enable them to move from one place to another in search for food, partner, or to escape from predator and threats. Both have a pair of muscle which work antagonistically, which means that when one muscle contract, the other will relax. Now, the differences is grasshopper have exoskeleton while earthworm have hydrostatic skeleton. Exoskeleton support and protect the body organ, enabling the insect to move, while hydrostatic skeleton maintain a rigid body shape of the earthworm. So one is to support and protect, one is to maintain the shape. Muscle in a grasshopper are flexor and extensor muscle, while muscle in the earthworm is circular and longitudinal. Grasshopper move by jumping or leaping, while earthworm is in a wave of peristalsis. They move in a wave of peristalsis along the body. Okay, grasshopper use leg while earthworm use kite. Kite is the bristle below the body to grip the soil, okay, or the substratum that they are moving on. Okay. So they anchor to the soil by using their kite. A student had a car accident and his femur was fractured. The orthopedic who treated the student had given him a new treatment technique by injecting a solution of carbon nanotubes at the fractured femur. The tubes serve as a scaffolding for the body tissue to grow. So this is how they use the carbon nanotubes as a scaffolding for the bone regeneration. So the bone will then grow onto the carbon nanotube, okay, right? So the broken femur then can be regenerated back and form back. So what are the advantages? They want 4 advantages and 1 disadvantage. So the carbon nanotube material used is very lightweight. So it doesn't increase weight to your body. It's very lightweight. The carbon nanotube material is high in mechanical strength, and its properties is the same as collagen, which is the main component of the extracellular matrix of this tissue. and it lacks toxicity. That means it doesn't cause the patient body to have toxin. So therefore, the patient does not reject the tissue. They don't experience rejection issue. So this allow the real bone to grow back and is biodegradable. That's very good, because after the bone has grown back, the carbon nanotube will just degrade. Okay. The other thing is it has high elasticity. That means it's elastic, and it is very durable. Okay, let's look at the disadvantage. Now, the cost of material is very expensive. The carbon nanotube is very expensive. You need expert to prepare the carbon nanotube solution, and you need high technology machine to operate and to do the carbon nanotubes. Okay. Let's look at the next question, question 11. Diagram 11.1 shows the Malaysian healthy plate menu with a concept called quarter, quarter, half. This concept is a quick visual technique that helps us to identify whether a dish is healthy and balanced or otherwise. So you can see that the plate is filled up with quarter chicken, which is the protein, quarter rice, which is the carbohydrate, half is filled up with fruits and vegetables. Discuss how the menu can help a person stay healthy. How can the intake of quarter, quarter, half make a person remain healthy? Okay, first of all, the quarter plate comes from carbohydrate, example, rice or bread. Now, rice or bread provide sufficient carbohydrate for energy requirement. However, if you don't follow the quarter, quarter, half, you eat excess of carbohydrate, it may lead to obesity, diabetes mellitus, or cardiovascular diseases. Next, the other quarter of the plate comes from protein in the form of chicken, fish, or egg. Sufficient protein intake is important for cell growth and repair. However, if you take excess protein, it may lead to obesity or even gout. The half plate comes from vitamin and minerals. Sufficient vegetable, fruits, and fiber is important to maintain a person's health and to prevent diseases. Fiber also helps to prevent constipation, hemorrhoid, and it also helps with peristalsis movement and stimulate bowel movement. So these are the answers for question, first question. Let's look at the next question. B, table 2 show the menu with recommended daily energy requirement of a pregnant woman and the menu with the daily energy taken by Amina during pregnancy. So Amina, who is pregnant, is taking food that is compared to the daily energy requirement, which is on the left side of the table. So if you look at the energy comparatively, for example, for rice, the recommended daily energy requirement is at least 700 grams, but Amina is taking only 200 grams. So you can compare the data between what is being taken by Amina as well as what is recommended daily energy requirement for a normal person. Okay, let's see the question. Based on table 2, justify the menu taken by Amina, whether it meets her daily energy requirement or not. Explain your answer. Okay, so if you look at the comparison just now, the values, seems that Amina is not taking sufficient quantity of food that can provide her with the energy requirement for the day. Okay, so the total energy intake does not satisfy a woman's daily energy requirement. Now, the total energy value intake is less than what is recommended. Her total energy value intake is 800 plus 640 plus 525 plus 630 plus 7,000 plus 504 plus 500 and plus 900. Now, this value you obtain from this side of the her daily energy requirement, which is on the right side of the table. Okay. You add up all these. So you get a total of 11,499 kJ of energy, okay, which is lower than the recommended daily energy requirement. Now, the reason why is because she takes less rice, less carbohydrate, less source of energy. Okay, she takes less rice, which provide her with the carbohydrate. That means she have less source of energy. She also take less bread, which is less carbohydrate and therefore less energy. She takes more butter, which provide her with a lot of fats, which can lead to cardiovascular disease. So Amina diet is not balanced. Okay, let's look at another question. Diagram shows a weight loss campaign to control obesity through gastric bypass procedure known as bariatric surgery. So the procedure is shown here, where part of the stomach that will be used is made from the entire stomach. So this part of the stomach is bypassed. Okay, so much of the stomach area is bypassed. So the portion of the stomach has become smaller and it is connected straight to the small intestine, which means that this part of the duodenum is not being used. It has bypassed this part. Okay, so justify bariatric surgery in obese patient. Discuss the advantages and disadvantages of the surgical procedure. So you can justify here means you can agree to the procedure or disagree, but you have to discuss the advantages. Okay, so in this case here, if you can say the obese person should or should not undergo the gastric bypass surgery. So these are the advantages if he does go for this gastric bypass. Now, the advantages is that if he goes for this bypass surgery, he will get fuller easier. Because his stomach is small, so he will take less food and he gets full faster. He gets full easier. Less calorie is absorbed because less food is taken. Okay, since he's already obese, this will reduce the amount of food that he takes, so he will definitely lose weight and become more healthy. This procedure also can help him to reduce blood glucose level, and it's easier for him to control his blood pressure. And overall, he will have an increased self-confidence level. So the final thing is that his body size will become smaller and his weight will decrease. Okay, the disadvantages are It can cause acid reflux because now the stomach is very small and it has bypassed part of the duodenum. So acid reflux means the acid will move upward into the esophagus. So this is the stomach sac that has become smaller. The actual stomach size is much bigger. It may easily cause vomiting. It may cause stomach ulcer, increasing and higher concentration of gastric juice. He may eat less, therefore he will obtain less nutrient. Less nutrient will be absorbed, which will lead to malnutrition. The length of his small intestine is shortened because the stomach bypasses the duodenum and connects straight to the small intestine. It is very costly to go for this procedure. It may cause bacterial infection because this is actually a surgery. There may be a risk of getting an infection. And the surgery can cause internal bleeding. Okay, so that is the answer for the disadvantages. Okay, let's look at the next question. Diagram 11.3 shows lymphoma cell of a patient. The type of food taken by the patient are important during the treatment of this disease. So this is the lymph node. So the lymphoma cell, which are the cancer cell, are formed inside the lymph node. Suggest a suitable diet that should be taken by this patient continuously over a long period of time. Okay, he should consume high protein or white meat. He should avoid red meat. Okay, he should focus on more on white meat so that it can quickly help to repair and replace damaged tissues. He should eat high-calorie food. High-calorie food is provided by carbohydrates to provide sufficient energy during cancer treatment. Okay, and a good diet will be able to provide optimum health condition. He should also consume a lot of green vegetable and fruits, which contain a lot of phytonutrients, mineral and vitamins to increase his immunity. And it also helps to stimulate the release of enzymes that can break down cancer. Okay, so this is the answer for this question. Okay, other than that, you can also suggest fruits and vegetables which contain high fiber that will help in digestion. Consuming orange color fruit which contain lots of carotenoids, the pigment carotenoids and beta carotene. To protect the body cell from damage caused by free radicals. He should avoid taking carcinogenic food that contain carcinogenic substances such as artificial coloring, artificial preservative, because this might increase the risk of him developing new cancer cells. He should avoid preserved food or flavored food because high salt intake can actually cause primary cellular damage that can increase the risk factor of a stomach cancer. He should avoid consuming red meat because red meat can increase the risk of bowel cancer. He should avoid taking alcohol because it can cause liver malfunction. Okay, With that, I end the discussion for this session on paper 2 MRSM SPM trial paper 2021. Okay, bye.